So the first thing I would like to do is to also let you know, I was given a note that I will state. So as all of you know, we have a global Angelman registry. Just so you're aware, which I'll talk a little bit more about later, it was one of the most exciting things the FDA learned about Angelman syndrome is that we have a global Angelman registry. And what they were also really excited to know is that we have such motivated parents that they're all willing to sign their children up. So with that said, we have now 826 families that have registered for the registry, which is amazing. So a round of applause to everybody who has done that. It takes 2 minutes to register, 2 minutes to register, but it takes over an hour to do all of the registry input, which you can do at home. But we have a challenge for all of you, and the challenge is that we want to hit 1,000 today. And there's a booth outside where you can go and register, or you can register on your phone. Megan Cross will be giving a presentation about the registry, and I want to make sure that you're all prepared to register, because what we have learned is we have an anonymous donor for everybody that goes and registers will donate $10 to FAST. So since there are 800 people in the room, I'm expecting that that's about 400 families, then that should be $4,000 donated to FAST. So every one of you that aren't registered, please go and register. It's very, very important for our community. With that said, I am so honored to introduce to you our first speaker, our keynote speaker, Dr. Timothy Yu. So many of you may know who Dr. Yu is because I'm so proud of him that I've posted him and his story all over Facebook. But those of you who don't, you're going to be inspired momentarily. He's an amazing individual. Dr. Yu is a neurologist and a researcher from Boston Children's Hospital. He's a graduate from Harvard. He completed his MD and Ph.D. at UC San Francisco and a neurology residency at Mass General and Brigham and Women's Hospital. He's an assistant professor at Harvard Medical School and an associate member of the Board of Institute at MIT and Harvard. His research effort spans from gene discovery for autism and other neurodevelopmental disorders, using cohorts of tens of thousands of patients to develop precision medicine for patients numbering as few as 1. He leads an ongoing clinical trial that he's going to be talking about today, which is being described as the world's first rapid turnaround fully personalized medicine, in which a group sequenced the genome of a little girl with Batten disease, identified her single unique mutation, and designed an oligonucleotide treatment for her. He launched this clinical trial to treat her just 1 year ago. So we're really excited to hear his story, and we're really excited to share with you how he has become a member of our community of Angelman syndrome at FAST.
Thank you, Allison, for that really kind introduction. So hello, folks. Good morning. Looking around the room, this is my first time meeting the great majority of you all. And so some introductions are probably in order. To introduce myself, I'm going to tell you a story. And I'll apologize in advance. It's not about Angelman syndrome per se, but I think it has implications for Angelman syndrome and many of the other rare neurologic diseases and conditions that we really care about. And I hope by the end of the story it'll be clear why exactly I'm so excited to be here and to join this community. So thank you. So I'm a neurologist and a researcher at Boston Children's. And a big part of my group's focus has been on trying to find out the root causes of neurodevelopmental conditions like autism, Angelman's, and other related conditions. And I have to say it's really remarkable how in the last 10 years, tools have advanced to the stage where, as a field, we're incredibly good now at discovering new genes, at diagnosing individuals with genetic disorders. But less clear, though, is how do we turn these diagnoses into real, practical, real-world treatments? And developing medicines is still this chasm that's hard to cross. And it still takes a long time, generally speaking. And so this morning, I'm going to share with you a project that's got me more optimistic than ever about our prospects for bridging this gap. So I have some pictures, and names to share, I'd like to introduce you, with the family's permission, to Mila. Mila, some of you may know from the media, is now an 8-year-old girl from Colorado. And growing up, Mila loved eating, skiing, hiking, climbing. She was a bright, social, talkative, and very adventurous little girl. But as she grew up, her family noticed some unusual and concerning things. At first, they were relatively small. At age 4, she began pulling her bedtime books close to her face, having trouble reading them. At age 5, even though she was super coordinated, her family noticed she would occasionally stumble and trip. But then around her 6th birthday, things quickly turned very, very serious. Her stumbling turned into falling. Her speech, which had been voluminous, now she began to slur her words and then completely lose her words. Her eyesight began to fail, and one day she was sitting watching her favorite movie, Frozen, of course, on television, and her father came and slipped a magazine in front of her eyes, and she didn't flinch. And they realized that her vision, which they'd been progressively worried about over the previous several months, had completely failed, and she had gone blind. She was admitted for an intensive workup at Children's Hospital, Colorado, and that just only revealed more concerning things. They did an eye scan showing retinal degeneration. They did a brain scan showing cerebellar atrophy. They did an EEG showing that she was having seizures, not yet clinically apparent, but visible on testing. And this prompted a very extensive lab workup, which after 2 weeks of intensive tests finally led to a diagnosis. It was one that really no one was happy to hear. So a skin biopsy was the clincher. It showed these unusual fingerprint-like smudges in some of her cells. And these fingerprints clinched a diagnosis of something called Batten disease. What's Batten disease? Well, Batten disease is actually a family of 14 different diseases. They're each very, very rare. And they're caused by recessive mutation, meaning 2 mutations, in key genes called lysosomal genes. Lysosomes are parts of the cell that are responsible for the recycling of cellular contents. And her particular pattern of swirls here, it looks like a fingerprint smudge, pointed to a subtype of Batten called CLN7. Now, CLN7 is ultra rare. It's a rare subtype of already a rare disease. There have been only 70 cases reported in the literature. The average age of onset for symptoms is around 3.5. And these cases unfortunately progress characteristically, as our patient had, to vision loss, seizures, motor decline, and unfortunately, pretty rapidly thereafter, a vegetative state, and then premature death by around 11 or 12 years of age. And unfortunately, this is one of the diseases of the many diseases that we don't have a treatment for. So this was really terrible news. Now, Mila's family is pretty remarkable, and not unlike many of the families in this room, they rallied. They took some time to adjust to this diagnosis, but by time I mean a few weeks. And the very next month, they launched their own foundation, because one did not exist for CLN7 Batten disease. They didn't have a built-in community. And they launched this foundation to take their story public, to educate people about this disease, to spread the word, and to raise money, to try and kickstart research that they hoped could eventually lead to a new gene therapy. But there was a wrinkle in this case, and that wrinkle was that she actually only had half of a diagnosis. What do I mean by that? Well, her clinical gene testing had only shown 1 mutation in CLN7. And CLN7 is a disease where you require 2 mutations. So where was the second hit? I mean, was this actually the right diagnosis? Even if she were able to, her family were able to start a gene therapy trial without that second hit, would she have even qualified for such a trial? Her geneticist, Dr. Austin Larson, told them, look, you folks need to go have whole genome sequencing. It's an advanced version of the clinical sequencing that's available. And it's been done obviously, by the Human Genome Project, but it's something which is increasingly available, but still quite difficult to obtain. And the question was, this was 2 years ago, where could she have this done? This is where this social media provider comes into the story. You'll all recognize the Facebook logo. And how many people in the room know what PMG stands for? So PMG is actually, it stands for the Physician Moms Group. And this is a group of individuals on Facebook who've gathered who are medically trained moms and they swap tips on medical referrals, on parenting, on balancing career and family. And as it turns out, my wife is a physician and a mom, and we have 3 kids of our own. That, of course, gives me the thinnest of possible excuses to sneak in a picture of our kids. This is Cora and Claire and Lillian, the 2 twins. But anyway, back to the story. And so on January 13th last year, my wife forwarded me this message that she had just read on the PMG group. And it said, "I'm hoping the power of PMG can help my dear friend. Julie and I were college best friends. Her 6-year-old daughter had just been diagnosed with Batten disease. There is no cure. They have localized one of the defects in the C11.7 gene, but they need to identify the second. Their doctors are recommending WGS, whole genome sequencing. If anyone can help in any way, we would appreciate it so much." So it turns out that whole genome sequencing is something my lab does quite often. We've been doing it for a long time. So that next day, I picked up the phone, spoke to the family, and we set in motion a plan to try and move quickly to provide them with some answers. 4 days later, we had consented the family. 11 days later, we had our samples in hand, and 1 month later, we had generated complete genome sequencing of her mother, her father, and her baby brother. 10 days later, we found something rather unexpected, and I'll show you what that is. This is what whole genome sequence data looks like. This is from Mila, her mother, and her father. And what you see is that when we sequence a genome these days, we sequence it in very, very small bytes. Each of these bytes has about 50 bases of sequence, A's, C's, G's, or T's. Sorry, about 150 bases, A's, C's, G's, or T's. And each of those reads, those bytes, is shown as a little gray bar here. And we were able to quickly identify her previously known CLN7 mutation, which was a little blip that's too hard to see here. This was actually a known mutation in CLN7 Batten disease, and we established that this was inherited from dad, so that was mutation number 1. So we began looking for a second mutation, this time from mom, and what we noticed was that there was an unusual cluster of reads here that was in a place where traditional, whoops, that's the story. Clinical sequencing really would not have been able to, it would not have shown up in traditional clinical sequencing. And when we looked very closely at the sequence, we found that there was evidence here of, okay, we're good again. Evidence of an insertion of extra DNA into this spot that wasn't supposed to be there. Now, I won't go into the details, but suffice it to say that there were actually fully 2,000 bases of extra DNA in this spot, and that we eventually deduced that this was an insertion of something that's going to sound esoteric. It's called a mobile DNA element, something called a retrotransposon. Now, what the heck is that? A retrotransposon is sort of an interesting thing. It's a jumping gene. These are genes that live in our genome that actually hop around. If that sounds a little bit disturbing, it is. And amazingly, half of our genome consists of these stretches of DNA that have hopped around over evolutionary time. Now, very luckily for us, the great majority of these are silent. They're like extinct volcanoes. But in some of these elements, though, are actually hot. And what had happened in Mila's family is that one of these hot volcanoes, these hot retrotransposons, had actually copied and pasted itself and landed in the CLN7 gene. Now, what does that do? Well, in this case, it had landed in this region of CLN7 that was in between 2 parts of the gene that normally code for the actual protein that we care about. Well, what impact does that have? The existing protein coding parts are fine. Well, we did something called RNA sequencing, and we found that the presence of this extra DNA in this spot was actually changing the way that Mila's cells were reading the genetic code. And now, instead of splicing from this protein coding region 6 to this region 7, now it was actually splicing from this region 6 into a new spot that it shouldn't have. And what that ended up doing was scrambling the sequence in such a way that the gene from that point on could no longer be read, breaking the gene in half and then causing her disease. So that was our answer, an interesting, academically interesting mutation, one that's tricky to find. It changed the assembly of this gene. And she had this plus her father's mutation, and that together confirmed her patient's diagnosis. The more we looked at this, though, we realized we could actually do something about this. There might be something more here. There might be a special opportunity. And that was that we could actually try to fix this. The idea here was that we could use something called an antisense oligonucleotide. I'll explain what that is. And we could block this abnormal splicing, this abnormal assembly. And this would, in principle, allow us to skip over this red misassembled spot and then allow it to restore normal connections between the exon 6 and exon 7, and then now restore function of this gene. And of course, we were only emboldened to think about doing this because the very same month that Mila was diagnosed, we were reading in the papers about the oligonucleotide drug nusinersen. Now, nusinersen is a drug that had been just approved by the FDA for another really terrible disease in kids. This is called spinal muscular atrophy. This is a disease where infants are born weak, and they just get weaker and weaker. And unfortunately, because of respiratory failure, they become too weak to breathe. They often pass away, typically around age 2 or 3. And what the FDA had approved was a new drug that actually changed the way that the instructions for this gene are assembled in a manner that actually was exactly like what we found was happening in our patient, in Mila. And that the success of this drug in clinical trials was so obvious that they actually stopped the trial early because it was working so well they could no longer ethically continue randomizing kids to the sugar pill. So we decided that we ought to try a similar approach for our patient. Now, oligonucleotide drugs, like nusinersen, are injected by a spinal tap. And they're injected, and this is the low spinal cord, this is the high spinal cord, and this is the brain. And when you inject it there, you find that it actually distributes all the way through, and it is absorbed by both spinal cord and brain. And so that was a promising thing. And when it's injected and absorbed, it's actually taken up by neurons very, very effectively. And if you look through sections of brain and spinal cord, you find that 90% of the neurons will absorb this drug. Moreover, when you inject it, it actually lasts a long time. As you can imagine, having a spinal tap doesn't sound like the most fun thing to have to do. But it turns out that this drug lasts for such a long time in the spine and in the brain that you can dose only 3 times a year, and levels will stay high enough that it's effective. So we asked, can we offer something similar for our patient? The parallels seem very, very clear. This disease, spinal muscular atrophy, was also a 2-hit recessive disease. It was also a disease that involved degeneration of the central nervous system. And they used an oligonucleotide with a particular chemistry that we figured we could repurpose and customize for our patient. We could give it by intrathecal or spinal tap administration. And we could use it to try to activate the CLN7 gene which had been disrupted in our patient. And so we began pulling every paper that we could about the 10-year drug development path that had led up to nusinersen, hoping that essentially we could accelerate that path and do essentially this. So having a template to follow, we designed the following strategy, and that was to generate cell lines from our patient. We would map this inserted DNA element very carefully, computationally identify critical hotspots that we could target with a drug like this, design and synthesize those drugs, introduce our oligos into her cells, and see whether or not we could get the gene to splice normally again. And we quickly found, our team was quickly able to make this work. So April, Jay, and Aubrey in my lab did something called reverse PCR from patient skin cells. And what we're seeing here in this picture, you see these two bands in this gel that's run here. The top band is normal splicing. The bottom band is the mutant splicing that our patient had. And what you saw, without any treatment, just patient skin cells, these were present at a ratio of 1 to 2. When we introduced some of our drugs into this dish and into these cells, we found that some of them were able to flip this ratio. You can actually see this here. This bright band on top is basically excess or boosted levels of the normal splice form that was successfully induced by application of one of our drugs. And so we did a series of experiments like this, repeated them multiple times, gave them to collaborators, investigators, didn't tell them what the drugs were, just said, do this experiment in a blinded way, and let's just confirm our results. And we were able to convince ourselves that we were actually able to do this in a consistent and potent way. This is an experiment that shows that treating patient skin cells boosted levels of normal splicing of the CLN7 gene from this baseline of between 7 and 11% up to about 3 times that. Okay, so that shows that the RNA is being corrected, but, well, are we sure that this is actually making the cells healthier? I mentioned that this is a defect in a part of the cellular process, part of the cellular machinery that's required for recycling proteins. And when you can't recycle proteins in this sickness state, what you end up are these accumulations of essentially cellular trash, these white bubbles here, shown on electron microscopy. And here they are blown up bigger. And in the green box are shown the effects of treating these same cells with our patient-customized drug. And I think you can see pretty clearly that these white bubbles are much reduced in their appearance. We did this and a whole series of additional experiments to convince us that her cells were really, truly getting healthier. And by all these measures, we were really gratified to see that it seemed to be working. So I'm going to pause here and recap, show you a little bit of a timeline. We're feeling that, well, let's see, December 2016, that was her clinical diagnosis. January 2017 was when we first met the family. February, we completed whole genome sequencing. In March, we identified this mobile DNA element as the second mutation. April, we generated cell lines. May, we had proof that her mutation was changing with splicing. June, we began to talk to the family. Hey, we may have something here. July, we designed a series of oligonucleotides. August, we began to have conversations with the FDA. Obviously, if we have something that's working on the bench, was there going to be a path for us to get this moved forward in a time that was in time to help our patient? September, we had evidence that our drugs were fixing splicing. In October, we had evidence that her cells were healthier. So we're feeling pretty good about ourselves at this point. But when we stopped and paused and talked to advisors, we realized that from a traditional drug development perspective, there's still a lot more to be done. We had really only covered step 1, proof of concept. And from that point on, there's still a lot more: manufacturing, release testing, formulation, testing in animals, and then phase 1, phase 2, phase 3 trials. It goes without saying that there's a real sense of urgency to do this fast. While we were working, our patient's neurologic function continued to decline. And her family sent a letter detailing all of these different ways in which she was losing milestones over the year that was elapsing. And she was still walking, but it was getting difficult. She would still smile and laugh, but in response to her family and to music. But that was beginning to happen, it seemed, to Mom less frequently. And so we said, look, we need to find a faster way. This also shows, traditionally, to get things through these next 3 steps, it's a multi-year process with a lot of money thrown at it. We began talking to folks and realized quickly that one possible avenue for getting something like this through the FDA was to open up what's called an individual patient expanded access investigational new drug application. And this is a route that's typically used for repurposing a drug that industry has made for one purpose and then using it for somebody else, for a different disease, a different indication. However, there was really no precedent for using this route for an entirely new drug customized for a patient. So we began making phone calls. We opened up conversations with the FDA. We identified regulatory specialists, and we found very, very quickly a really critical team of advisors, including Lauren Black, who's sitting right here, and you'll be hearing from her later this afternoon. Mary Pendergast, Sarah Goldkind. These are all ex-FDA individuals who had a lot of good advice for us about how to proceed. We also wrangled an invitation to an industry symposium and found experts from industry and manufacturing, testing, formulation, toxicology, all of these remaining pieces that we need. And in collaboration with all of these folks, we set out to tackle the challenges that laid ahead. There were plenty of challenges. For example, let's look at manufacturing. So the problem is that there's really no applicable FDA guidance for a rapid turnaround case like this. And just from a manufacturing perspective, making the drug fast and safely is the most critical step. In our case, our needs were a very small batch of this drug, 10 grams, that would be sufficient for a lifetime supply for our patient. But when we called around to manufacturers and said, "I'd like 10 grams, please," they laughed and they said, "Well, that's 50 times less than we normally do. We start at half a kilo. And it would take 6 to 9 months, and it's going to cost about $500,000. Right? So we had to call lots of folks and ask for people to bend some rules for our support of our case. And we found an outfit that was willing to do this on a small scale with rapid turnaround, yet still with the quality that we needed. Together with all our advisors, we put together a very aggressive timeline. Perhaps the fastest attempt at getting a drug from an idea into a human that has been seen yet. And in principle, this was a path to get us from manufacturing, animal testing, and release testing into a patient trial in as little as 4 months. We made arrangements for the manufacturing of a patient-grade drug. This was not trivial, getting the manufacturing and then shipping to multiple outfits and then testing, shipping more, bottling, more testing. There are a lot of moving parts to coordinate. And getting this done could probably be a talk in itself, but suffice it to say that with the help of folks listed here on this page and a lot of heroic efforts and weekend time spent in the synthesis labs, we were able to get it done. On December 12th, our partners successfully manufactured a single clean lot of drug for our patient, and we named it, fittingly, Mila after our little girl. Of course, there was one more wrinkle, that the drug had been made, but then the facility called and said, "Hold on, before we can ship it, there's a little matter of some legalities that we have to address." And I'll share a little bit of that with you. Lauren is smiling. And that was there was a labeling arm wrestle. So we had originally agreed that our drug was going to be made and it was going to be called for research use only, because it reflected the fact that we had taken some shortcuts in the traditional manufacturing of this drug in order to get it done in time. And they called after having made it, and their lawyer said, "We're not comfortable with that anymore. We want to call it not for clinical use." And we thought, well, that doesn't make any sense. We plan to use this clinically. We're going to go and take it and inject it into a patient. We said, well, how about we call it investigational use only? And they chewed on that for a couple of days, and then they came back to us and said, well, no, I don't like that. How about we call it research grade? And I said, come on. We've got a schedule to keep. We've got a patient waiting. We have animal tests to run. Okay, why don't we call it investigational use limited to this particular IND application? Or heck, why don't we just label it for Mila? Because that's what we're going to do with it. And they sat and we sweated that out for about 36 hours. And finally they came back to us and they said, look, this is what we'll do. Research grade material limited by US law to intended use as stated in IND 136294. It barely fit on the label around the vials to get all of that in. So just a little flavor of the kinds of new challenges that we had to address. So finally, after a 1-week legal standoff, the drug was finally shipped we said, please take a picture of it. I want to see it leaving the loading dock. So here's a picture of it being shipped out. And it left the loading dock in San Diego. The very next day, it arrived in the hands of Julie Duville, who's sitting at the table right here, from Charles River Labs. Her team was responsible for now taking this and doing our animal toxicology tests. And they got started the very next day. And we started a toxicology program designed by our colleagues at Charles River, intrathecal injections in rats at 3 different doses, careful neurologic exams by rat neurologists, and pathology exams. And then we examined these animals at 1 week, at 3 weeks, and 3 months. We developed a clinical protocol for our patient. We would dose by spinal tap. We would start by gradual dose escalations, treating every 2 weeks. And then we would extend the dosing interval out to 3 or 4 months. We had careful safety monitoring around each dose. We had exploratory outcome measures, nothing nearly as well thought out as what was presented at ABOM yesterday, but we didn't have time. On January 13th, with all this happening, the family, in preparation for a possible treatment, moved to Boston and arrived in the snowy weather. We actually hadn't received permission to proceed yet, but luckily, 6 days later, after looking at the early rat data, which looked good, the FDA said, yes, you may proceed with your trial. Well, we still actually hadn't received the drug yet, but that was being done by a team in Florida. And once it cleared sterility testing, we received 2 precious boxes of our drug on January 25th. And 6 days later, we launched our trial. So how are we doing, and how is Mila doing? So we are about 11 months in. And first, safety. So I'm very happy to say each of our doses has been well tolerated. There have been no drug-associated adverse effects to date. What about response? Is it working? Well, we should say this up front. This is a compassionate use trial. We have an N of 1. There's no placebo control. There's no crossover period. It's still relatively early in the course of this disease. But I have to say we're seeing some very promising signs. We know that in the year before we started the trial, Mila's disease progressed in several different domains. That letter from the parents showed that. And in the 11 months since we've started the treatment, our clinical observations are consistent, and our parents' reports are consistent with the idea that we haven't seen anything decline any further since we've started. So it seems that progression may have arrested. Of course, we'll know more in the months to come. What about actual data? Those are our impressions. Well, here are some of the measures we're tracking. And neuropsychologically, we're looking at a number of disease rating scales, neuropsychological development measures. And what we're seeing is that these are, over this short time period, are relatively flat. What I do want to point your attention to is the seizures. So just 2 months before starting treatment, Mila started having 30 seizures per day. And these were pretty intense, lasting 1 or 2 minutes at a time. And they're quite debilitating. And what we see on this graph here is that over the course of the trial, her seizures became much, much less frequent. And at one point, they actually got down to a nadir of 0. They've bounced back a little bit. They're usually around 0 to 10. And more strikingly, their duration, which used to be 1 to 2 minutes, now has gone down to a few seconds to perhaps 30 or 45 seconds at the top, but mostly just a few seconds. And her family describes that when people come to visit, they almost don't notice them. The seizures are so mild. So this actually is quite striking. It breaks the rule for Batten disease, which is that seizures are expected to get worse and worse and worse. And so we think this is the most promising sign that our drug is really having an impact and helping our little girl. So let me leave you with a few thoughts and then now come back to Angelman syndrome. So for some types of mutations, it's now conceivable to go from a diagnosis to a customized drug in just a year's time. And it's got us thinking, can we actually scale this? Can we imagine a future where there are rapid interventional genomics units offering custom-made drugs for patients with life-threatening diseases? We're thinking of this might be possible, and we're recruiting patients to try and pilot this idea right now. Second, this case has been a really wonderful reminder of how important it is to keep working on models of collaboration between researchers, hospitals, patient foundations, industry, and the FDA. And when you've got everybody aligned, as you do in your program here, and pulling together, you can really do amazing stuff. Three, our treatment happened to be individualized, but it provides a proof of principle that has broad implications for many other treatments, including some for Angelman's. And our experience gives me great optimism for the use of antisense oligonucleotides as a platform for developing these kinds of genetic therapies. It's a powerful tool and a really exciting time. So I've taken the long way around in introducing myself to you all, but I hope by now it should be reasonably clear why I'm here. In March of this year, I got an Allison from Dr. Berendt, Allison. She introduced herself first as the mother of Quincy, a 3-year-old girl with Angelman's. Second, as the chief scientific officer of FAST. Third, as an expert veterinarian along with her husband. And fourth, as the chief operating officer of GeneTX. And she said that GeneTX wanted to develop a first-in-world treatment for Angelman syndrome, and that it was going to be an oligonucleotide like Milasen, and it was going to be supported through angel investors that are also parents, and that they were aiming to get this done as fast as humanely possible. And she asked if I'd be interested to help, and of course, how could you say no, right? A couple of days later, I was on a call with Paula, Jennifer, Allison again, Scott, Debbie, a number of other folks. And I did say, look, I've been studying autism and neurodevelopmental disease a long time. I do have to say, I'm not an Angelman expert per se, but I'm willing to learn. And I've also got friends and colleagues like Dr. Wenhan Tan in my same division at Boston Children's Hospital, who is a card-carrying Angelman expert. And I've been following with admiration the animal work and the genetics of Angelman syndrome done by people like Art Burdett and Scott for a long, long time. So after meeting this great team that Paul and Allison have assembled, I said, I'm happy to help in any way that I can as an advisor for your drug development program and to share our experience with Milasen to help prepare for a clinical trial. Later in the day, you're going to be hearing an update on the exciting oligonucleotide strategy that's being developed based off of proof of concept by Scott and Art Burdett. And this is using an ASO to reactivate the paternal UBE3A gene. I won't go into any of the details because I don't want to steal any of that thunder. I'm running a few minutes over, so I apologize. So I'm going to stop and close with a few thoughts. Okay. I do have a couple of cautions. Speaking to remote parents, I do want not to get overexcited. I am extremely optimistic and extremely excited, but a few cautions just worth noting. ASO therapies are not risk-free. In animal models, there are things that we see, like inflammation or nerve damage and so forth, and so these experiments do have to be done carefully. Caution number 2, accelerated development, as in Milasen, in under a year is probably not yet the new normal. Doesn't mean we aren't trying, but it is worth saying an N of 1 compassionate use trial is different than a new drug application for a drug that's going to market and going to be given to thousands of patients, right? So there are additional considerations that you have to put in play that we got to skip in Milasen. And there are also additional considerations for neurodevelopmental conditions like Angelman syndrome that I've been thinking about for a long time. Conditions about finding the right treatment window, when to intervene, and so forth. But those cautions aside, it's early days for ASOs, but really good reasons for optimism. Nusinersen, Milasen, a couple of other drugs that are either in humans or actually another one, patisiran, which is actually FDA approved. These are all ASO therapies that are looking really, really good in the market or in at least investigational trials. And so I hope that pretty soon there'll be another to add to that list. But look forward to working with you all towards that goal. So I'm just going to stop there. I'm going to put up a slide with acknowledgments to many people who helped us in our Milasen work, bringing in expertise from well beyond my lab and well beyond my institution, but from industry and patient advocacy and regulatory and so forth. It really takes a village to run these projects, but you've got a village here, so I'm real thrilled to be a part of it. Thank you.
I think we have time for maybe 2 or 3 questions. There are microphones on the side of the room on each side, and we have runners, so raise your hand as well if you are far from a microphone.
Art Baudet, I really congratulate you on the spectacular work. I wanted to just ask, how did you convince the FDA to forego primate studies?
Right. Really good question. So what I was impressed by in our conversation with the FDA was really how open they were to discussing something which was relatively novel to them. I think that they haven't seen many cases like this. And one thing they kept stressing was that it's all about the risk-benefit, right? And on what we had her clinical decline on one hand, we had in vitro data on the other hand. We also had some limited resources too, right? And so we were really asking ourselves, would we learn more from a small monkey study or a beefier rodent study? They gave us actually the allowance to go with a single species as opposed to the usual 2. And we actually made the call ourselves that we thought that we would actually learn more from a dosing perspective and just from a sheer numbers perspective from doing the rodents. Right.
Yeah, I mean, there's a precedent for Lauren Black from ex-FDA, and there's a precedent with, for instance, when you look at drugs that are designed for an infectious disease, and there's absolutely no target for that targeted medicine in the animal, you can just do rats. So we sort of recapitulated that argument and said that the exon skipping mechanism that was actually going to be workable in Mila was not actually part of the genome of the monkey. And so we weren't going to get relevant pharmacologic data in the monkey, so a rat was just as good. Do you track with me? Did I answer that question? Okay.
Go ahead.
Yes. Hi, Christina Alberini, New York University. First, wonderful talk and wonderful data. I have a scientific question. Do you have data on, do you restore the normal level of the normal splicing form? Or, I was gathering from your information, or do you degrade the long splicing and the whatever is expressed from the other correct form remains the same?
Right. Thank you, and that's an excellent question. So in our experiments, we were able to show that we could triple the amount of normal splicing in our case, that the level at which it was not 100% rescue. However, because this is a recessive condition, we know that carriers of CLN7 mutations are perfectly fine. They don't show any symptoms of this terrible disease. And so we knew that at least 50% gene activity was sufficient. So we took advantage of that fact. The fact that it was a recessive disease meant that a little bit of gene restoration could go a long way. And so that gave us the confidence to proceed, even though we weren't getting 100% restoration.
Last question. Go ahead, Sib.
So I guess my question is, while we all think that there are barriers in the FDA, did you deal with any barriers within your own institution in really leading this and pushing this forward, certainly for any potential side effects or adverse reactions?
Well, I will admit that the first response from our legal team was, "You're trying to do what?" And certainly there is a lot of exposure when you give a novel agent to a patient like this. But I have to say that after sitting and reviewing this case with our ethics team and leadership all the way from the top all the way down, I'm really proud to say that Boston Children's decided that this was the right thing to do, and that they were also gutsy enough to allow it.
Great, thank you, everybody. He's going to be here all day, so feel free to bombard him at lunch.